starts off straight away with a security alert. Yeah, so I'm going to talk a little bit about the work we've been doing on deeper coral reefs. And so there will be a bit of genetics in there too, but I like to call it molecular ecology, so it scares off less people, I guess. Yeah, so first to start off a bit and put things into perspective, usually when we're talking about coral reefs, we're obviously referring to the first tens of meters of the water column, and usually have the association with these type of reefscapes, or at least in the Indo-Pacific. But I think it's good to point out that this first 30 meters really only represents a quarter of the depth extent to which scleractinian corals and tropical reef communities occur. So this zone is called the Mesophotic zone, as many of you know, and but still with a lot of people this brings the association with large gorgonian fans and sponges, and it's good to point out that we can actually get really quite significant coral cover still at lower mesophotic depth. So this is an example from Curacao between 75 and 80 meters with really high coral cover. And then in the case of the Great Barrier Reef, we find scleractinian corals with zooxanthellae all the way down to 120, 125 meters. So this is really a significant proportion of the depth range of coral reefs, not just on the Great Barrier Reef, but in the world. And in terms of the spatial extent, a lot of these reefs are obviously still undescribed, and we don't really know that much about them. But it's estimated that in case of the Great Barrier Reef again, they can occupy a similar surface area as that of shallow reefs. So actually really quite significant in terms of their spatial extent. There's obviously a very good reason that we know so little about these deeper reef stills, and that's just because they're hard to access, and not to the same extent as deep sea ecosystems, but it's sort of this annoying in-between depth range where we still need some of these tools, such as ROVs, submersibles, and technical diving to actually access these depths. And so we've been lucky enough with this Catlin Seaview Survey to establish some of this infrastructure and do some research. And so what I'm going to present today is mostly with examples from the Southern Caribbean and Bermuda, even though we've been doing a fair bit of work as well on the Great Barrier Reef and the Coral Sea. So other than the spatial extent, why are we interested in these mesophotic coral ecosystems? Well, initially a lot of this interest was actually driven by the observation that deeper reefs can sometimes be protected from common shallow reef disturbances such as storm events and warm water bleaching. And so the idea was that for species that are shared between both the shallow and deep reef, that they might be able to find refuge in these deeper reef habitats during disturbances, and then aid in the recovery of shallow reef areas by providing propagules. And so really a lot of this initial interest and also funding was driven by this instrumental value of deep reefs to shallow reefs basically. But over time, as we've been working more on these deeper reefs, we obviously get confronted as well with some of the unique biodiversity that you find in these deeper reefs. And so there's also been a growing interest in the intrinsic values of these deeper reef ecosystems. And so over the past couple of years, our main overarching questions that we've trying to get close to getting some proper answers to is figuring out to what extent these deeper reefs can act as a refuge in terms of how much biodiversity really is shared between shallow and deep reefs. And then for species that are shared, so depth generalist species, is there in fact vertical connectivities? Is there movement of larvae between deep and shallow reef areas? And then in terms of intrinsic value, I mean, I've obviously referred to some of this unique biodiversity, but how much unique biodiversity really is there? And can we talk about a specialized community, particularly lower mesophotic depths, or is it a marginal extension of the reef slope? And in case, if it is a specialized community, what's the vulnerability of these communities? Usually when we talk about deep reefs, it's in the context of being more protected, but we know about disturbances that occur over large depth ranges. We know of disturbance specific to deeper reef areas, such as cold water bleaching. So yeah, figuring out how they, to what extent they can cope with such perturbations. And so we've been using a molecular ecology approach to address some of these questions. So first of all, using phylogenetic methods to really get into that biodiversity question, largely because it's an issue that's not simply resolved with taxonomy, particularly working with deeper reef specimens that inherently look different from what you find in the shallow. And on the other hand, using population genetic techniques to obviously get at this connectivity issue. So both vertical connectivity as well as horizontal connectivity, so between different mesophotic reefs. And so the first challenge, obviously, working with mesophotic reefs is the whole access thing. But the other issue that we had to overcome with this specific product is just the general lack of good markers for corals. And so some of the commonly used traditional markers for coral phylogeny, they fail to even distinguish some of the well-known species, and some even can distinguish between well-established genera. And so that's a real issue, obviously, if you want to look at phylogeny at this level, or biodiversity at this level, I mean. And so in terms of population genetics and connectivity, obviously the tool of choice has been microsatellites. And while this is a very appropriate tool, the issue we've had there with corals is that they have a relatively long development time, particularly because they have symbionts. And so even though we've been interested in these questions of vertical connectivity over the past 5 years at least, we've only seen studies for 3 species in that time period. So obviously, specifically the whole resolution thing is something we can easily overcome now by sequencing entire genomes, which become more affordable, but we're still talking about several $1,000s, and that's obviously not something we can do to really address some of these questions where we want to sequence hundreds to thousands of individuals. So we've been using a technique called restriction site-associated DNA sequencing, but basically, simply put, it allows us to sequence the same random subset of the genome for a large number of individuals. The only issue with this technique is that we're still working with corals, and so they're obviously in association with symbionts, and if we extract DNA of those, because it's a non-specific technique, it just focuses on restriction enzyme or other repetitive sequences, depending on the exact method you use. And so when we sequence those, we get a mix of both, which you could think, that could potentially be actually quite interesting, except that if you don't have a reference, these are anonymous reads. And so you basically get a combination of the two, and there's not much you can actually do with that data set. And even if you would have a reference, given a larger size of the genome for Symbiodinium, it's actually really quite hard to get good coverage for your coral loci. And so first part of the project was to really sort of develop this methodology and get this working for corals. And so in short, how we do it is that we basically physically try to separate as much as possible the coral and Symbiodinium prior to extraction. And then we actually isolate out the Symbiodinium using flow cytometry based on cell size and fluorescence. So we can sequence the Symbiodinium separately, and we use that as a subtraction reference. And so we use a technique called NextRed, which is developed by Eric Johnson at the University of Oregon, which is actually a primer-based approach, so we can work with smaller genomic DNA input, and then sequenced on HiSeq, NextSeq, but I won't go into too much detail. And then obviously we had to develop the bioinformatic pipeline for the separation of those reads and the analysis. And so we've used this now for about 1,700 corals that we individually sequence, both for the connectivity population genetic component and the phylogenomic component. And so basically that's allowed us to now move quickly and look at different species to really sort of get a better grasp on what's happening with vertical connectivity on the reef across a number of species. And so in the Caribbean, we've focused on Egerecia lamarcki, Egerecia fragilis, and Stephanosoni intercepta. And in the Indo-Pacific, we've worked with Pexir speciosa, Cropra aculeus, and Ceratopra hystrix. So these are some of the most common real depth generalists that you find in these 2 regions. And so instead of going through each individual one, which has its own story, I'm just going to try and summarize a bit the general pattern that we've been observing. And I'll do that with the help of 2 of those different species. And so this is a structure plot, so basically showing individuals on the x-axis and then the probability of belonging to a certain genetic cluster on the y-axis. And this is for populations sampled around Bermuda. And Egerecia fragilis is a brooding species. And basically what you see here is that there's strong genetic differentiation between depths, so 10 meters and 40 meters, but not between different locations, except for the one on the end, the green cluster, which is the most western population. So strong genetic differentiation between the depths, but not locations. Then on the other hand, if we sample those exact same populations, but for a different species, Stephanosoni intercepta, which is a broadcast spawner, we don't see any significant structure at all. So both well connected across depths as well as different locations. And so even though we find species and geographic difference, so within a species, sometimes we see vertical connectivity in one location but not in the other, the emerging pattern is that for brooders, we actually quite commonly find genetic differentiation, whereas broadcasters are the most likely to actually have vertical connectivity. And that obviously also makes sense in terms of the reproductive mode. The other thing I'll point out here is that with 4 out of this species, we've run into cryptic diversity issues, which a lot of people that work on coral population genetics and such run into, but it does highlight, that is a real issue with corals. There still is so much undescribed and cryptic diversity out there, which is annoying as well for these kind of assessments because it reduces your population sizes. So that's talking about vertical connectivity, so that's mostly about the upper Mesophotic reef and the shallow reef, where we still find a lot of species that they have in common. But we're also interested in those lower Mesophotic communities. And so this is a case study that we've done in the Southern Caribbean, Curaçao and Bonaire. And this was really to address the question whether the communities below 60 meters are a specialized community or not. And that was mostly driven as well by the fact that the marine park is actually defined by the 60-meter isobeth there. And so we were quite interested to just figure out what's below that boundary. And so we did these initial surveys using the submersible, and initially we saw that there was only 2 dominant species that make up this community, or so we thought. And so that's Madrassus forensis, Agricia grame. Agricia grame is a depth specialist species, but Madrassus forensis actually occurs in really quite high abundance on the shallow reef as well. So, I mean, are we talking about a specialist community here? Not really. But we also did a lot of sample collections, and so we sequenced the Madrassus as well. And so this is again using that the red-seq method, so we can actually look at tens of thousands of loci or concatenate them all, and basically found that the deep Madrassus forensis is actually completely distinct from the shallow one, and in fact represent a distinct species that is a depth specialist, because we haven't found it on the shallow reef. Then on the other hand, with Agricia, the same thing, so we did a lot of red sequencing there as well. And so this is originally with traditional markers, we couldn't even pull apart the different well-established species, but now with this red-seq we can. And what we found there is that large part of the Agricia grames that we collected actually turned out to be Agricia undata, which we could already see before sequencing. But Agricia undata was supposedly rare or non-existent around Curaçao, but it actually, a large part of the lower Mesophotic community consists of this Agricia undata, and so sequencing confirmed that as well. And then for the Agricia grame, we actually found that there's 2 sympathetically occurring lineages that are genetically quite distinct from each other, and so likely we're dealing there with 2 different species as well. So in fact, we're actually dealing here with 4 species that are depth specialists, and so I think we can really say that we're dealing with a unique, specialized community here. And so obviously we were interested because it occurs below, okay, that was the video. But I guess this shows it well. This is what a community can look like between 75 and 80 meters, where you can get really quite high coral cover, but often there's these sort of large monostands. And so we're interested to look a bit more into the vulnerability of these communities. And so one of the first things we did was doing these growth experiments, transplanting the corals between 60, 80, and 100 meters. And one of the interesting observations there is that actually the growth rates at 60 meters were comparable to those that we observed for Agricias in the shallow. And so obviously we're talking about thinner skeletons, so the amount of calcium and carbonate that's being put down is definitely not the same, but in terms of linear extension, it's actually really quite similar. And we also sampled 9 different locations around Curaçao and Bonaire, so just these deeper water communities of this deep specialist species. And although we found no clear genetic differentiation or clustering between the different locations or even the 2 islands, which is quite positive, we did find really quite high levels of clonality within these communities. And so the other thing we did is to actually, within these monostands, we mapped some of those out and actually genotyped different parts of those monostands. And it turned out that some of these plates that are within 5 or 6 meters of each other usually represent the same clonal genotype. Okay, so just to summarize, obviously this red-seq approach has really helped us progress some of these questions a lot more quickly, just because it skips that development phase. It gives us that increased resolution. In terms of connectivity, we can also look beyond that at signatures of selection, etc., which I won't have time to present now. but it also really demonstrates that phylogenetic potential for a lot of outstanding questions that we still have, particularly at this within genus level, so shallow phylogenies. Vertical connectivities, we're finding species and geographic differences, so it's really hard to make broad sweeping statements about the role of deep reefs and shallow reef recovery. It's a case-to-case basis that we have to evaluate this on, but we are finding this general pattern that it is more likely for broadcast spawners. In terms of the lower Mesophotics, this was just a case study, but for there we did find a specialized community that does warrant management consideration. And this is obviously work that we're now extending to the Great Barrier Reef as well, where issues of biodiversity are a bit more complex. And vulnerability, it's obviously very early days because it's just from a single species, but for this specific species it looks like growth rates and connectivity are favorable, but we are finding high levels of clonality, which it is worrying in terms of the genetic resilience of these communities. And obviously this is not work that I've been doing just by myself, there's been a lot of different people involved, so a big thank you to them as well. Yeah.